Okay, hi guys. Okay, so this is our enzyme, and hopefully we are all really sure that this is catechol oxidase. So we milked that potato, produced this enzyme, and it is really a protein. So it's a polypeptide, and what we mean by that is it's a bunch of amino acids and they are linked together with peptide bonds. So we've talked before about the structure of a protein, just a little bit in a previous video, but we just want to remember that a protein, this is its primary structure, just a bunch of amino acids grouped together, one by one, linked together, and that is its primary structure. But the trick to these things is that they don't just fold once, okay? So this primary structure really does look like a string of beads. They're just stuck together, strung along with these peptide bonds, just beads on a string that you laced a needle through. That's exactly what this looks like, and that's really not going to fall apart because these are covalent bonds. But some of these are polar, and I'll just put that, and some are non-polar, so I'll put NP. So that makes it fold differently. So what happens with our secondary structure? Here, we'll go secondary. As we start folding up our molecules, and the one thing that happens first is it chooses either a beta sheet structure, and that's supposed to look like a folded piece of origami paper there. And that's called a beta sheet. And then it can also have the option of forming an alpha helix. And that's just a single spiral. So when you think of DNA, that's a double helix. We just have sort of a corkscrew shape here with the single strand. So the alpha helix is going to fold because there's going to be polar and non-polar repelling or attracting each other. And so then you get a molecule that's kind of convoluted, and there will be some beta sheets, and we'll just try to put some in there. That's a beta sheet there. I know it doesn't look like one, but that's okay. And then we'll put some alpha helixes over here, and maybe another beta sheet over here. And it's still all part of the same molecule. We'll just try to make that look more beta sheety. So as it folds up and it becomes more globular, that's the tertiary structure. And each one of these, you can be polar or non-polar, and all the polars, so you can be polar, but you can have a positive or negative charge. So you're going to get along with water, and the positives are going to attract each other, or to the negatives. Or another positive plus positive is going to repel each other. So that is another complex interaction that determines how this protein folds. Now, why is this important? It seems very complex and convoluted. We want to know that when we get to this part, when we're attracting positives to negatives, these are weak partial bonds. So they're partially ionic bonds. So because it's not a full positive and a full negative attracting each other, it's really weak and transient. So when we took our enzyme and we boiled it, we basically broke this structure down to its just maybe secondary structure. And if we did a really good job boiling it, we get down to this primary structure. Now when you break those bonds, if you break down this tertiary structure and you put it down in a simpler form, either the secondary structure or the primary structure, it doesn't have an active site. This basically melted down to just be a plain old brick. So it doesn't have an active site. Active site is fried. So we took our catechol oxidase and we boiled it for a really long time. And so we're breaking down this structure either to this form or to the primary form. So that's probably where we were at. And it's just, can this guy grab onto catechol and oxygen? No. So there's no products. No products at all produced. So hopefully that makes sense. Those were all the experiments that we did in the Enzyme Lab. So there is a lot to go back and look at. And if you still have questions, be sure to let me know. I hope this helps. Thanks.